Okay, then I would like to continue with myself. I would like to present data that we've generated with Shizuoka Cancer Center in Japan and with the Dutch Cancer Center here in Amsterdam. So I'm presenting also data from Yasuhiro Ko, Dr. Ko from Shizuoka Cancer Center. To sketch a little bit the context, well, we all are aware of the fact that cancer is a leading cause of death, that it is increasing. Fortunately, there are new approaches. And these new approaches are being discussed today over here. These are sometimes called the golden bullets. Well, that was 10 years ago. Now we've found that maybe these are golden bullets, but you have to point into the right direction. You have to identify the patient that is expressing the target, but also in a way that can be approached by the bullet effectively. If we talk about lung cancer, it's, well, the chance of getting lung cancer are unfortunately pretty high. Here are the numbers for the males, numbers for the females. What's the best treatment? Well, that's an easy one. If you look at the population, stopping smoking. As you can see here, and the most interesting discovery, I think, in this field, as far as I know, has been made not by molecular, not by a biologist, not by a chemist, not by an oncologist, but by a statistician. It was an English statistician who found this correlation and could prove the correlation between smoking and death by cancer. And when that was identified, when he made this, when he presented his data, you immediately saw that there was a drop in the number of deaths caused by cancer. So there's an easy golden bullet, and that's stopping smoking. The other alternatives, as we talk about chemotherapies, then in the old days, that was when I was 4, the combination chemotherapies were being used. Nowadays, as we will be talking about more, the targeted therapies are there. Like gefitinib, one of the early compounds developed against the EGFR kinase receptor that was mentioned a couple of times. You really see that the overall survival has increased. I will talk more about the EGFR receptor in a minute, because it's very much mutated in particular tumor types. And it was already mentioned in rectal carcinoma. But if we talk about lung cancer, then between 10 and 30% of the patients harbor this mutation in this receptor. Well, what's its job? The job of the EGFR receptor is to detect growth hormone. So if the hormone binds to the receptor, it starts signaling by its intrinsic kinase activity. That kinase activity is a kind of way to transduce the signal to the nucleus where all the proteins that are needed for proliferation, for example, are being generated. And that can lead to tumor growth. So generating all this information by the molecular biologist has resulted in an inhibitor. I'm going to talk about Tarceva, erlotinib, which is an inhibitor that directly binds into the ATP, a crucial factor for a kinase, the ATP binding site. And by blocking the kinase activity, it blocks the transduction of the signal from a growth factor, asking the cell to grow or to differentiate, into a signal activity. Interestingly, there's also this protein, KRAS, that was also already mentioned. So this is a kind of model, and also this molecule plays an important role in the transduction of this signal. If you would block over here, but the signaling continues here because there's a mutation, for example, then it's in vain. You can block whatever you want. But if signaling is starting over here, then you're not working at the right side. So in this field, people are looking at EGFR mutations and KRAS mutations. Why EGFR mutations? Well, that's because if you use the inhibitor, gefitinib, an EGFR kinase inhibitor, you can clearly see, and these plots have been introduced to you already, here the time, months since randomization, for example, as progression-free survival, starting with 100%. You see here the result with standard chemotherapy, and here the result with gefitinib. So patients live longer, but in the end, most of them still die. So I want to present some data on DNA mutations from the Shizuoka Cancer Center, with whom we collaborate, and particularly Dr. Ko, who already mentioned. And this is his plan. This is what he's working on. He takes a tumor tissue from a patient, and this tumor tissue is being used in standard pathology for the hospital, for the medical decisions, but also in his research. In his research, he's using lysates for kinase profiling, DNA for mutation analysis, and also total RNA for transcription analysis. And if he looks at these tumors from lung cancer patients, and he starts comparing, and he presented this in Sapporo last month, smokers versus non-smokers, and you look at the mutations in these patients, you can clearly see that EGFR plays an important role. At least it's detected in many of these patients, 11%, but much higher in the non-smokers. I also would like to draw your attention to all these other observations being done. Also BRAF, which is a kinase which is just downstream of the KRAS molecule I just mentioned. The same pathway. PY3 kinase mutations, EGFR amplifications, c-MET. So it's a plethora of defects that you can encounter when you start analyzing the mutations in such patients. If we compare the two types that are being observed, the adenocarcinoma and the squamous carcinoma, again, EGFR specifically in the adenocarcinoma mutated. But I think what's very interesting to see as well, and it was already touched upon by Giorgio, a lot of patients have, there's no molecular information available at the DNA level. And also remember that at the DNA level, it can be, you can find a defect if you can find a mutation, but whether it's a relevant mutation, that remains to be seen. Whether that mutation is druggable is the next step, and whether that drug is capable of inhibiting that particular defect activity is the big question, especially because other activities can play an as important role. The EGFR. Of course, I should also mention the most interesting new molecules being investigated. ROS kinase. ALK was mentioned by Giorgio. It was identified in lymphoma. But here, about 4 to 5, 6% of the patients also express this mutation and are now being treated with crizotinib very successfully. And the new kids on the block, the RET kinase, a very important neurotrophic growth factor. DDR2, well, and more will come, as I said, because a large percentage of the patients has an unknown mutation or maybe no mutation. So Dr. Ko is very much interested in this kind of molecular profiling of patients. And as I said, he's also using these lysates for protein kinase profiling. And the next I want to show you is some data that we generated with the NKI that really made Dr. Ko enthusiastic to have this type of analysis now standard in his research. Well, what's the kind of missing link? Well, there's a lot of research still to do. But as we said, DNA abundance, well, let's focus on DNA mutations. It can identify a defect gene. But that's a weird word. A gene can never be defect. Its function is to get into a translated protein. And that works well, also with a mutated gene. The problem is that the translation from a DNA mutation to an RNA presence, maybe there's a mutation in a gene that's never expressed. Well, and it's not that relevant to investigate. But if the protein is not expressed, we have the same problem. And in the end, we would like to look at the activity of the protein. The activity of the target class. For example, the activity of kinase or nuclear receptors or phosphatase or what have you. And we would like to use more and more these clinical samples that fortunately by the oncologist are being collected more and more. For example, to use in these xenograph models that Giorgio talked about. An exciting possibility of using these activity-based assays is that you can investigate also the effect of the drug. Because a drug, like a kinase inhibitor, it doesn't act on RNA expression. Maybe in the end, remotely. It does not act on the protein abundance itself. No, it acts on the kinase. And the function of a kinase is to transfer a phosphate from ATP to a protein, to get that protein active. So we would like to measure where the action is, you could say popularly. And the advantage of such an activity-based assay is you can also add the drug into your assay, into your test. And that's what I show over here. So here we took a sample from a particular patient or a cell line or a tissue. Just the extract is made, is put onto the chip, and you see these different spots. These spots represent different peptides. These peptides represent different proteins, and these proteins are part of all these different pathways that already have been mentioned. And if you then add the drug on an adjacent chip or an adjacent array, you can see that some of the activities cannot be inhibited, others can be dramatically inhibited or intermediately. And that's because the phosphorylation of the peptides on the chip are affected by the drug directly, which is artificial, which is, of course, ex vivo, but at least it's information about the drug engaging, often for the first time, engaging in the lab the target as in the way it's expressed, the kinase, the way they are expressed by a particular patient. What we do more and more, and this is an example in a totally different field of renal cell carcinoma, where we are investigating what is the effect of, for example, sorafenib and sunitinib on the kinase in a particular patient. I show this slide also to explain to you what all these kind of patterns are about, because we're not all familiar with heat maps, possibly. Each line here represents a peptide on a chip. In the previous presentations, it can represent a gene on a chip or any other kind of readout. And the more blue it is, the more intense blue, the more effectively it was inhibited, this particular peptide, by the indicated drugs on the chip. So here we see peptide phosphorylases being inhibited, also there. So sorafenib and sunitinib are the same in these, similarly capable of inhibiting phosphorylation of these peptides in the tumor of this. This is a single patient, also here, also there. But if we look into the normal tissue of this same patient, and we start comparing then sorafenib with sunitinib, then sunitinib is a very dirty compound, rather dirty compound, if you compare it to sorafenib. Well, this type of experiments we call pharmacology on chip, and it shows you that we are now investigating what is the effect of the drug outside of the body on the chip onto the kinases from this particular patient. Even kinases not in the tumor but in the normal tissue. We're using this in the Framework 7 program to identify also markers that can predict the toxic effects of the drugs, tox markers. The workflow is straightforward. Samples are being taken and either frozen or you can directly use it. But most of the times we work also in this lung cancer program with samples that are from archives or prospectively collected, frozen and defrosted, lysed for 30 or 60 minutes, then analyzed in our instruments. We get the signals on the chip. How are we applying this? Well, if this is a timeline where a patient is being treated with, for example, a kinase inhibitor, and at this point you get a biopsy. This is the ideal point. In the data I will show you, we were not that lucky yet. We had to work with later material. But ideally you get biopsy material over here, and you generate a molecular profile. The molecular profile is then being used to identify differences between responders, clinical responders, and clinical non-responders. But that information is most of the time not related to the drug. It's a molecular profile, but on a sample that has never seen the drug. So what we like to do is to generate these drug response profiles ex vivo, because this is drug-related information, drug-rich information. And we hope there's a higher chance of identifying then activities and inhibitions thereof that correlate to the clinical response. In other words, does the on-chip response predict the clinical response. So now I will show you the data that we generated with the NKI, with Dr. Hauke Klomp, Professor Baas, and a lot of work has been done by Riet Hilhorst, my colleague who's also here, and will present later today on another topic. As I said, lung cancer, here again the molecule Tarceva. I already explained to you, using these mutation detection methodologies, you can identify the patients that express this particular target with the mutation. That can be targeted with Tarceva. Patients with these mutations are being treated. But there is already information in the literature showing that also wild-type patients, well, at least patients that do not harbor this particular mutation, can respond to therapy. That was the aim of this trial, in which they, in a neoadjuvant setting, started treating patients with the drug, and were wondering whether also the wild-types could respond. Why? They were using a PET scan technology and CT scans in order to see if there are responses. But a molecular approach was chosen to look at the activity profiles, the kinds of activity profiles. Why looking at these? Because you can already look at the mutation of the EGFR and the mutation of the KRAS. But as said, we want to have higher level data, at least related to the drug. So we started spiking in the drug indeed into the lysis of these patients. And another interesting way of thinking is that if you want to identify markers that predict response, you should also identify markers that predict non-response. Non-response is resistance, and there's more and more data in the literature showing that other pathways not related to the target pathways are responsible for this non-sensitivity, this non-response, this resistance. I call these XFR because they are all PDGFR, they are IGFR, CMET, AGFR. But if these are active, then again, you can start blocking whatever you want. The tumor still gets, the cell still gets the signal to proliferate, and the drug will not be effective. So markers predicting this, markers monitoring this, are maybe a way to go. So that was the aim here. In a preoperative setting, so that's called neoadjuvant therapy, patients were treated with erlotinib, and these activity profiles were used to identify a prediction marker. I should say a classification marker, but I'll get back to that later. In other words, this is the whole population. There are no approaches, if these patients are wild type, to identify a possible responder or non-responder. And we set out and used the approach that I just explained to identify maybe a class, a molecular profile, activity profile, to differentiate responders from non-responders. This was the setup. So on day 1, patients got the treatment with erlotinib, the pharmacotherapy, till day 21. Then there was a washout period. At day 24, the tumor was taken out, and resection material was used for kinase profiling and for pathological assessment. As said, preferably we'd like to have worked with this material at the diagnostic stage, but that's an interesting approach for the follow-up experiments. How does it look like? Well, I explained these heat maps with either high inhibition levels by the drug or low inhibition levels in blue, and we could clearly see differences between non-responders and responders. This is what I always like about science. Totally unexpected result. Because we said that maybe the responders on the chip, in the clinic would also show on the chip response, but it's totally the opposite. We see here that especially the non-responders show high kinase inhibition response, while the responders less. Still show response, but less. So maybe this can generate a new hypothesis on the mechanism of action. This is a difficult picture, but I'll explain. On the y-axis we have here the score, a prediction score based on the PamChip data, based on the activity profiles. And it's either positive or negative. It's a complicated algorithm that's being used called partial least square discriminant analysis. And if I can try to explain it, if you have so much data for example, 144 readouts. If you plot these 144 values in a hyperdimensional space, like a 144-dimensional space, they get a certain location in that space. If you do that for the responders and for the non-responders, then you can draw a plane that differentiates these two groups. Then you're with a new sample. You do exactly the same experiments. You get again 144 readouts. You plot it in this space. You're just going to check whether it's on this side or on that side of that plane. And the distance to that plane determines whether it's a positive value, positive prediction value, or negative prediction value. Well, Rick DeWine, he was already mentioned. He can explain much better than I can, but this is kind of the basic principle. So we get positive values and negative values. If we compare that to the clinical data, in blue the non-responders and in red the responders, you can see there is a correlation. There are also some differences, but let's first focus on the clinical data, because we also have here some information on the mutation status. For example, EGFR, the asterisk, this circle identifies the patient with the KRAS mutation that I explained. But these are the most interesting ones. Patients that do not have this EGFR mutation would have never been selected for this new targeted therapy, but they do respond in the clinic. They are red, but there's no marker because there's no asterisk or no circle. Using this profile, we have a first candidate marker because we could, as I said, using this data, determine a prediction score which was negative, indicating it is a responder. So this was a very exciting observation. Then we asked our collaborators from the NKI 13 new samples. We analyzed these again on the chip. We had to add Tarceva. If we did not add the drug itself, the data were not correlative to the clinical responses. We used this prediction model, and again, positive values and negative values. Then we asked for the labels, whether these patients were observed to be clinical responders or non-responders. And again, we see a clear separation, a correlation between the score on the chip, negative or positive, and the result in the clinic. And again, here 1, 2, 3, 4 patients that are wild type that do respond, but would never been selected for therapy. So in summary, before I go to the summary, I showed you only signatures, only these heat maps, without any annotation of the peptides or of the proteins from which they have been derived. Of course, there is more information that can be derived from that, but it's not straightforward. Nonetheless, if we do this type of analysis, we just compare the responders to the non-responders statistically and identify the peptides that are most discriminative, significantly, then we identified, for example, that a peptide derived from a phosphorylation site in the platelet-derived growth factor receptor was involved. Well, this is an interesting new hypothesis. We call it a hypothesis generation exercise. Maybe this is what's happening on the chip is also happening in the tumor cell. Well, we can use all kind of classical methodologies like Western blot, immunohistochemistry, etc., to confirm or to reject if needed. In summary, so also wild type or non-mutated patients could be selected for therapy. Maybe I should go back one slide. So let me reiterate that these patients do not have the mutation, but maybe they have another mutation. So this also generates hypothesis. Maybe we should start, and that's what Dr. Ko is going to do, maybe we should start analyzing these patients on on the complete sequence of EGFR to see whether there are other mutations than the ones that we have been focusing on. There is some good evidence that kinase activity can at least correlate to the pathological assessment, being a responder or a non-responder, and therefore it's a fingerprint for the non-mutated patients. As said, it's immunohistochemistry from the start was not possible. As said, there was no marker, but using now the data, and maybe we can extract some pathway information from that, we can determine which kind of immunohistochemistry test should be tested, should be investigated. And I mentioned this only if we use this experiment of spiking in the drug with the patient-derived material, this classification was possible, otherwise not. But as said, we should be critical. The material we used could not be really be used for prediction, because it's taken after the patient has been treated. But Rick did also some analysis comparing patients that have not been treated with the ones that have been treated, and then it was impossible to get significant differences if you build such a model that I just explained. Another interesting discussion point is that maybe in these 21 days, the tumor cells that are affected are just gone, and maybe the ones that remain are not completely different. They possibly don't have a completely different wiring, and that could be a rationale that also the results we obtained using these post-dose samples would also work with the pre-dose samples. But as I said, that's food for discussion. In summary, in lung cancer, we're looking at DNA, we're looking at mutations of EGFR, fortunately of more and more and more targets. With this approach that I explained, we try to expand the field where we can identify these markers. I borrowed this from Bertrand Jourdan, one of the chairs of the next sessions, in order to increase the chance of identifying markers. And as I said, not only markers that are related to the target pathway, because often there is information available, but of the resistance pathways. And from the start, that's a big unknown in many research programs. We involve the drug and we test the real target. And this may be a jump to the next session, translational research, where the targets are being investigated in model systems. Finally, I would like to thank a lot of people in Japan, the team headed by Yasuhiro Ko. People from the Netherlands Cancer Institute already mentioned how Klomp and Paul Baas, but especially Eva Schaake, was a crucial factor in this whole study. And the complete PamGene team, not only the R&D team, because a lot of people have been involved, but Reed Hilhorst and Lisbeth Haukes, especially, many thanks for their help in this study. Thank you very much.